Good morning or afternoon, everybody. My name's Elisa Hill-Yardin. I'm from RMIT University in Melbourne, Australia. Today I'll be talking about glitches in the gut-brain wiring, understanding gastrointestinal issues in autism. So my team's particularly interested in the Gut-Brain-Microbiota Axis, and we're really keen to work more on this interface between the enteric nervous system, the second brain in our gut, and microbes. So this tells us a lot about the interface between gut content and the gastrointestinal mucosa. We also want to know more about the interaction of the enteric nervous system with bacteria, and this leads, of course, to understanding neuroimmune interactions and how these types of interactions may be altered and potentially contribute to gut dysfunction in neurodevelopmental disorders. So we're particularly interested in autism spectrum disorder. And today I want to talk a bit about aggressive behaviors, which can become a major concern for families and people living with autism. I'm going to focus mainly on gastrointestinal symptoms and some information on neuroimmune interactions. We know that there are a range of gastrointestinal symptoms that are common in people with autism, and these can include alternating diarrhea and constipation, bloating, abdominal pain, reflux and vomiting, for example. And this is really important. We now know that people with autism are much more likely to be hospitalized for gastrointestinal problems as well. So one of the models that we're particularly interested in is the Autism-associated R451C mutation in mice in the neuroligin 3 gene found at the synapse. So we know there are a whole host of rare genetic mutations, which we've heard about previously in this session. We can group them together by their function, and we know that several of them are involved in neuronal communication or keeping synapses the synaptic structure intact. And so the neuroligin family are shown here in red, and many others that genes encoding these synaptic proteins that have been associated with autism are highlighted in red here as well. So the neuroligins are postsynaptic gene adhesion molecules. The mutation we're interested in is a missense mutation, changing an arginine to cysteine residue at position 451 in the neuroligin 3 protein. And it was first identified in two brothers with autism spectrum disorder. And both of them we found with the in a collaboration with Christopher Gillberg and Maria Rastam in Sweden and Thomas Bougeron in Paris, that both these brothers actually have gastrointestinal dysfunction, and one of the brothers has problematic aggressive behavior. So the GI symptoms that we have that are on the clinical records for these brothers include esophageal inflammation, fecal incontinence, regurgitation, diarrhea in one brother, and also the other brother showed chronic intestinal pain and diarrhea. So when we look at the mice, we do know that there are, if you like, glitches in the brain wiring of the mice. We know from the NL3 R451C mice first reported in Science in 2007 by Thomas Sudhoff's group that there's an alteration in various brain regions in terms of the excitability, the neuronal activity in these mice. They also show behaviors that are relevant for autism spectrum disorder, so they're a nice model. We, of course, wanted to know, do these mice have aggressive behavior or heightened aggressive behavior, and did they show any gastrointestinal dysfunction? So when I first imported the mice to Australia in 2009, I was working with them, and I noticed that they were incredibly aggressive, and they were fighting a lot more than what other mice that I'd worked with before. And in order to test whether this was related to the mutation, we conducted a territorial aggression assay. And so to do this, we put the test male, either a wild type or a NL3 R451C mutant, in an isolated cage for 1 to 2 weeks and didn't change the bedding so that they can emphasize their territorial area. And then we introduced a junior male of another strain and assessed the time to attack, number and duration of attacks, and other behaviors. So all mice show territorial aggression, but we wanted to see whether this was more extreme in the mice with the autism-associated mutation. In fact, it was. If we look at, highlighted in green, and we look at these clear boxes, you can see that this steep curve shows that there were a higher percentage of the mutant mice attacking within the first 5 minutes of the assay compared with the other groups. So the wild type is shown here in the dark grey. And this was the same for the increased number of attacks by the mutant mice compared to the wild type, and also the duration of attacks was higher. So we also gave another group of these mice an atypical antipsychotic, which is commonly prescribed for irritable behaviour in children with autism, and that is risperidone. And we saw here that this was able to bring down that aggressive phenotype to wild type levels, and this is shown here in the red bars. So overall, we were quite astonished to find that the R451C NL3 mice are 8 times more likely to attack than their wild type litter mates. And we've also repeated this. This was on a mixed strain background. We've now repeated this on a Black 6 background in our 2018 paper. So, of course, leading on from this, we wondered whether the synaptic mutation could affect the neuronal activity in the gastrointestinal tract and perhaps contribute to gastrointestinal function, as seen in the brothers with this same mutation and in a large population of people who are living with autism. So to do this, we needed to study the enteric nervous system, and as you know, it's a second brain. It really is a complex nervous system. It has at least as many numbers of neurons as the spinal cord and a whole range of the neurochemistry that we see in the brain. So there are 2 main neuronal plexuses between the muscle layers all along the gastrointestinal tract, and the first here is the enteric plexus, predominantly responsible for regulating gut motility, and then the submucosal plexus, more involved in regulating transmucosal movement of fluids. And so, of course, we needed to check whether or not neuroligin 3 was expressed in the gut, and it is. And this is work done by Shani Herath, who's just submitted her PhD thesis, and she did some very nice developmental work optimizing RNA-scope for the gastrointestinal tract and 3D reconstruction of neuronal types, showing very clearly that neuroligin 3 is expressed in this example, the mouse submucosal plexus in the ileum. And so leading on from that, we also wanted to know whether the expression was altered and whether neuronal populations might be affected in the gastrointestinal tract in the mutant mice. And so we found, interestingly, in this example, overall in the ileum submucosal plexus, that there was no change in neuronal numbers, but we did find increased numbers of vasoactive intestinal peptide neurons and decreased numbers of cholinergic neurons, as shown here. But in fact, we've seen in different regions along the tract, we've seen significant changes in the neuronal proportions just due to this missense mutation, and only subtle phenotypes in other ways in these mice. So next, it was important for us to see what functionally might be altered in terms of the gastrointestinal tract. And to do this, this is very nice work done by Kevin Moo, who did a 9-month project in the laboratory. And we first looked in live animals using X-ray imaging. And so these mice are fed barium sulfate and then imaged at various intervals, and we can track the bolus of barium sulfate throughout the gastrointestinal tract. And we could see, really quite surprisingly, that there was a very clear difference in the transit time. So the mutant mice have a much faster transit time throughout the entire gut, but also when we analyze different sections, each region showed faster transit time as well. So you can see up the top here in this example, for a control mouse, you can see that the fecal pellets containing barium sulfate are produced much later. So the transit is faster here in the lower row, showing the mutant mouse, where the fecal pellets are emerging about 40 minutes before that of the control. So this is great, but we then needed to move to an organ bath assay, because, here the brain is attached, and we know that the gut and the brain are communicating all the time. So perhaps this could be just an effect of a change in anxiety or something that is centrally nervous system mediated. So what Kevin did here was that he used the video imaging approach, where we take a segment of gut. It's put in physiological saline at 33 or 34 degrees. we video imaged the contractile patterns, and we were able to convert them to spatiotemporal maps as shown here. So each red streak is a neurally mediated contraction starting at the proximal part of the colon and ending in the distal. Now, we've already published that in the mixed background that these mice show a subtle change in motility, which is GABA-A receptor mediated. However, these experiments were done on a black 6 background, and we again showed a GABA-A receptor mediated change in gut motility. It was a subtle change where these short contractions that we see in the distal colon are reduced in the NL3 mutant mice. So there we have, in the whole animal, we see a change in gut transit, and we also see, as a result of the enteric nervous system, changes in the gut contractile patterns in the colon. So moving on from there, we wanted to know whether perhaps the submucosal plexus might impact mucosal permeability in these mice, and we know that changes in the nervous system activity can influence the epithelial barrier, and increased permeability can lead to changes in immune responses and potentially gut symptoms that we see in patients. So this part of work was done in the ileum in neuroligin 3 and wild-type mice, and we did in fact see, using an Usen chamber approach where we measure the current, and we first measured the flow of FITC dextran particles from across a segment of gut. We found that the neuroligin 3 mice do show increased permeability, and they show decreased transepithelial resistance. This is in the ileum, and we saw varied results, and this is unpublished, but we saw varied results at different regions along the gut. And this was work done by Shani and Herath. So this leads us to ask, well, do these mice perhaps have altered immune responses? And so we have had we have done some work on this, and we've chosen to look at the cecum, in fact, in a bit of detail in these mice. So the cecum is the equivalent of the human appendix. In the human, it's quite small, but in mice, you can see here it's actually larger than the size of the stomach. It's located between the colon and the ileum, and close up you can see at the apex of the cecum within here, when we pin out this region of tissue, we see these lymphoid aggregates, which are known as the cecal patch, and they contain a whole host of immune tissue, and they're important in sensing bacteria, microbes in the gut. So it's really not been known what the cecum, the function of the cecum is, but now we're starting to think that, in fact, it may act as a safe house or a store of our complement of bacteria, of microbes, and that when we get an infection and all the microbes are flushed out, for example, by diarrhoea, that we can then recolonize the gastrointestinal tract from this safe house store of microbes. So Samiha Shana, who's just finished her master's degree in the master's thesis in the lab, looked at this gut-associated lymphoid tissue in the cecal patch. She also looked at the nervous system and found changes there in the neuronal proportions, both in the myenteric and the submucosal plexus. But when she looked at the macrophages in the NL3 R451C cecum, interestingly, you can see the wild type across the top row and the mutant on the bottom. She saw increased density in this tissue. Macrophages were smaller, and they were also rounder in shape. So this suggests that perhaps there's an altered immune cell response to bacteria in the autism model. At the same time, it's important to mention that we have also been looking in the brain of these mice, and this is work done by Samantha Matter during her master's thesis, and she found an increased density of microglial cells in the dentate gyrus, and this is without an additional immune stimulation. She also found some subtle changes in astrocytes in the dentate gyrus as well. So looking further, when we look at the microbiome itself, we have some preliminary data showing that there's quite a large difference in the cecum between wild type and the mutant mice, and at different regions along the gut as well. And this is work done by Samiha Shana. As well as that, Shani optimize the protocol to assess the density of different microbial species, different microbial populations adjacent to the gut mucosa. And she did this study in the ileum, which corresponds with the increased permeability that she found, and she did find altered microbial density in these regions. So she looked at the phylum Bacteroides and Firmicutes, and she's also found some differences in Akkermansia muciniphila. Now, prior to this, we did compare the feces, and we found altered microbial populations, and this was in mice that were housed in mixed cages, which is very important. We saw a cage effect on the microbiome, but within those cages, we could still see microbes that were present only in the mutants versus microbes present only in the wild types, which tells us that there is an effect of the genetic mutation. So to wrap this up, where are we with this? We're looking at the altered gut-brain axis in the neuroligin 3 model of autism. We've got altered synaptic proteins. A lot of work has been done in the brain in this model. We've also shown altered microglial characteristics. The behavioral work we published showed an aggression phenotype and increased repetition. But what I've really talked about here is this gastrointestinal dysmotility, a change in permeability, and perhaps a dysregulation of the immune system, which is something that we're working further on. And we also showed dysbiosis. So our aims going forward are to further understand this interaction between the nervous system and the microbiome, and perhaps be able to modify the microbiome to better understand neuroimmune pathways to promote gut health, and via the gut-brain axis, potentially improve mood and behavior as well, which would be a relevant findings for the population, for people with living with autism. So I'd like to thank the people who actually did the work. This is my lab here. Kevin and Samiha are past students. We have a joint laboratory with a group run by Professor Ashley Franks, who is an environmental microbiologist. And so a lot of our students spend half of their time looking at the microbiology. We have a number of terrific collaborators, and this is our funding, both past and present, for the work that I've presented today. Thank you very much.
So thank you for a wonderful talk. So as questions come in, maybe I'll start by asking you what you think what do you think the contribution of the immune system may be? And so in your model, you see intestinal permeability, which suggests that there may be bacterial translocation that activates the immune system. Is it you're thinking that that immune response may be a driver of either the GI or even the behavioral phenotypes, or potentially a consequence of dysregulated enteric nervous system function?
Yeah, great question. Chicken and egg, right? So, I mean, as a neuroscientist, I'm coming from the nervous system mutation first and foremost, and actually, I'm kind of surprised that we're seeing these microbial effects. But I think that there's some very nice papers now looking at mechanisms. Their function alters the immune system, and then that also changes the permeability. So these are papers from Anne Littman's lab and Gabrielle Bell's in Nature Immunology this year. So I think that, we've got this constant bidirectional communication that we've really got to learn a bit more about, and perhaps apply next in our model this inflammation insult and see what happens there.
Yeah, I know these things are the directionality of how these different systems talk to each other are difficult to convolute or deconvolute. What do you think in terms of the microbiome? So the changes in the microbiome, again, maybe the same question in different contexts. How much do you think they're contributing versus potentially being a consequence of just altered gut physiology? Have you done like transplant experiments or antibiotic treatments to see, is there a functional role for the microbiome in your model?
No, so we haven't had an opportunity to do those kind of experiments yet, but I wonder if what we're seeing is an amplification of something that is already altered based on the mutation. And this is why we're wondering if we can alter the microbiome, we can dampen the effects without obviously changing that mutation, but we might still see improvements. That's kind of the way we're thinking at the moment.
Okay, that's great. That was a great talk. So I was wondering, so those animals are litter mates, presumably, right? So are they housed together? I mean, just following up in Sarki's question, could it be that now if you were to change the ratio, or is the microbial changes that you see responsible for some of the phenotypes in your animals?
Yeah, so that's a great question. So in the fecal study that I was talking about, these were absolutely litter mates, and there were we could see the cage effect, but these were mice born in the same litters. There was just a smaller study, and it was very clear that we could still see an effect of the mutation within those mixed cages of the litter mates. So that was for the feces that we analyzed in the 2019 paper. With the unpublished data, it's a large number of animals in each, and I have to go back and look at the litter mates, but they are litter mates, and they're collected at roughly the same time. There's about 25 mice in each group, which is, quite significant. So I don't know. I can't really answer your question, but
no, because we also saw that in the Jean-Friby model, which is quite remarkable in terms that despite the animals are litter mates and they're in the same cage, you can still see a change in the microbiome, because usually we believe that when we combine a wild type and a mutant, now everything will be sort of the same. So it means that the selective pressure that you have there is quite pronounced, because despite the fact that or maybe they're in Australia, your mice, they don't eat poop. Maybe it's the other possibility, right? So
they do.
But that indicates that the selective pressure is still there and is quite robust. But yeah, that's good.
What I can say though is actually those separation of signatures in the fecal microbes were more obvious earlier on. If you sample weekly, you see it reduce, which fits with the coprophagy.
So Lisa, there's a question from the audience. Any metabolomics data on the R451C mice? And also, if so, do you think that metabolites may play a role in permeability?
So no, I don't know of metabolite data in the model. And yes, probably a lot of things play a role in permeability, really a lot that I think we're not really conscious of at the moment. So I definitely think that the metabolites would. And then as a knock-on effect of changing the microbial populations, that's obviously going to change probably the mucus biofilm that again can exacerbate any differences that you're seeing.
Yeah. And what about the biogeography? So I know you showed using the Using chambers that there seems to be increased permeability in the ileum, if I remember correctly. Where do you think the neuroligin mutation, where in the gut do you think neuroligin mutation may have the most effects, and does that in any way impact either a GI or other symptom?
Yeah, so that's an interesting thing. I think we're starting to realize that different regions of the gut have, well, we do know that they have different functions, right? But I think we're not yet investigating the effects of mutations in this way. But we certainly do see the ileum is more strongly affected in the data that I've just shown you. And I have heard that this might be the case in other models of neurological disease as well, in particular in the ileum, but I don't think that data is published either. So maybe that's a kind of a hotspot that we could be looking at.
Yeah, no, you're absolutely right. Is just over the past few years, we're learning more and more about what different regions of the gut are actually doing. And so I think that literature, again, still nascent in mice and humans, may guide you through additional physiological effects of these mutations.